Okay, hello and welcome everybody to another speaker event from us with Munich NLP. Usually we have guest speakers that directly talk about NLP and its applications, but today we have a very special guest who uses NLP and sequence processing techniques not to analyze natural language processing or natural language sequences, but to model amino acid sequences. Amelie Schreiber is a deep learning engineer from Los Angeles and holds a master's degree in mathematics, which she now uses to advance the field of protein language models. Her work focuses on refining these large models with techniques like low rank adaptation and their quantized counterparts, which are better known as LoRA and QLoRA. Applications for these areas in biology include predicting the function of proteins, binding sites for proteins, as well as post-translational modifications. She has also written amazing blog posts about these topics, which are available on Hugging Face, and we will definitely link those as well in the video recording, in the video description of this recording. If you now ask yourself what some of these terms meant, like binding site prediction and protein functions, or also what they have to do with our usual NLP methods that we discuss in this group, don't worry, this is exactly what this talk will be about. So with this, I will now stop talking and hand over the stage to Amelie. Thank you very much for being here and presenting today.
Yeah, of course. Thank you for having me. I really appreciate it. It's really great to be here with everyone. So I'm going to have sort of two different parts to the talk. The first part, I'm just going to talk about some different applications of protein language models. And then the second part, I'll talk a little bit about fine-tuning with LoRA and QLoRA. So let's just jump right in. There we go. So let's talk about the background a little bit on what these things are. Protein language models are language models like you know and love. They can be autoregressive or bidirectional. So you can think like ChatGPT versus like BERT or Roberta. The model that I'm most used to and that I'll be talking the most about today is more like BERT and Roberta than it is like ChatGPT. It's a bidirectional model. It uses rotary position embeddings, and it has a standard vocabulary of 20 amino acids instead of, like 50,000 English words or tokens or what have you. We just have the 20 standard amino acids in the vocabulary, along with a few non-standard ones, and a few extra tokens for things like indels, which are like deletions and insertions. And so the vocabulary is a lot smaller. The vocabulary for ESM2 only has 33 tokens in it. And it can be used for protein folding. That's one of the most interesting applications, I think. But there's a lot of other use cases that I want to cover today. Excuse me. So ESMFold is ESM2 with a folding head on top, and it can be used for protein folding just like AlphaFold 2. The only difference here is we don't use multiple sequence alignments anymore. We just have a single sequence go into the model, and we're able to get these really nice protein folds like this for protein sequences. And because we don't need multiple sequence alignments for protein language models, they end up being a lot faster. They end up being about 60 times faster. The accuracy ends up being pretty close to AlphaFold 2. For shorter protein sequences, I think ESMFold ends up being a little more accurate than AlphaFold 2. And for longer protein sequences, AlphaFold 2 ends up being more accurate than ESMFold. But there's a lot of overlap in between where they have very similar results on how accurate they are for protein folding. But like I said, protein folding is just one of the things that we can use these for. And I want to talk about some other applications outside of protein folding because they have a really large amount of use cases. Let's talk about a few of those. So some of the use cases that I have listed here, and I'm going to go through each one of these in detail, but we can score mutation effects on proteins. So if I take a protein and I mutate it and I want to know what the effects of the mutations are on the protein and its functioning, I can use protein language models to score them. I can compute something called evolutionary velocity, which is a kind of directionality to evolution. So if you have two proteins and you mutate from one to the other, you can get a score from the protein language model telling you whether that mutation was beneficial or not. And if that mutation was beneficial, you get a direction, an arrow directed from one protein to the other that tells you that evolution kind of flows in a particular direction. And we can do this for large amounts of proteins and multiple mutations and get a kind of vector field for the mutation landscape. And I'll talk a little bit more about that in a minute too. We can also fine-tune these models for various different things, like predicting binding sites, post-translational modifications. We can also do sequence level prediction tasks, and I'll talk about fine-tuning a little more in a bit. We can use them as the backbone in diffusion models, which I think is a really interesting project. There's not a lot of work on this, but there should be. And I'll talk a little more about how we can use diffusion models in a pipeline to construct binders. We can use them for directed evolution. So directed evolution is when you take a protein, you pick some mutations using a model like ESM2 and evolve the protein to optimize it for higher fitness or higher function. After we've done in silico directed evolution on them, we can predict protein-protein interactions, and we can fine-tune ESM2 for predicting PPIs. We can use ESM2 for computing intrinsic dimension. So intrinsic dimension, I'll talk a little more about that later, but you can think of it as kind of a stand-in for perplexity for masked language models. And we can also replace sequence search and do homology detection, sequence clustering, and replacing MSAs with protein language models. And I'll talk a little more about that as well. And also, structural models are often really important in things like drug discovery and docking and predicting how small molecules and proteins dock with each other. And we can use the embeddings from protein language models like ESM2 to improve the graph neural networks that we use for things like this. So if you use the embeddings as feature vectors in your structural model, you'll get improved performance this way. And I won't spend a lot of time talking about this because this is the one thing on this list that I haven't really worked a lot with. But I do know that a lot of people are interested in structural models, and they do end up becoming important, especially in like when you have small data sets, they end up being more effective than some of the larger models. And so I'll talk a little bit about that, but I won't spend a lot of time talking about structural models because I haven't worked with them that much. So this is just a heat map of the log likelihood ratios that we get from the model. So if we compute the log probabilities using the logits from the model, and we compute the log likelihood ratios between a wild type protein and a mutated protein, we can get a nice heat map here. And if you look along the bottom, this is just a segment of the protein along the bottom of the heat map here between residue 40 and residue 70. And then we have 20 standard amino acid mutations that we can mutate to on the left side here. And this heat map just tells me which of these mutations along the left side, are going to be beneficial or detrimental to the functioning of the protein. So if you notice this dark blue band in the middle here, this is telling me that there are a lot of mutations in this region that are going to be beneficial to the functioning of my protein. And if you look at the yellow hotspots, you can see mutations that are detrimental to the functioning of the protein. And I've constructed a little web app on Hugging Face Spaces that you can play around with that lets you do this for any protein. So maybe check that out, go play with that a little bit. And this is just explaining that again. So here the blue is blue and the red is the yellow. So here in this new diagram, red is the same as yellow for the last diagram. And this is just, again, showing like different mutations that end up being beneficial or deleterious to the protein's functioning. And in this particular case, we have fluorescent activity. So if we pick mutations where the dark blue is occurring in the heat map, we pick mutations that have higher log likelihood ratios, we actually end up getting more fluorescent activity from the protein, which is really interesting. So the protein language models idea of fitness of the protein highly correlates with the actual function of the protein. So we can use this to predict which kinds of mutations are going to be beneficial and which ones are going to be deleterious to the functioning of the protein. And like I was saying, we can get a directionality to the evolution. So if I have two proteins and they differ in some places, I can compare them and compute their log likelihood ratios and get scores telling me which direction evolution flows. And I can go from this local picture to this more global picture. And this is actually taken from one of the ESM papers. And this is not my diagram, but it's a really good illustration of what we get. And you can get this kind of like vector field picture where you see like a flow of evolution happening, where each point in this diagram represents a protein, and you see the black arrows are telling us which direction evolution is flowing across the protein landscape. And this can be used for things like predicting viral escape and studying evolution of different kinds of proteins over very long time periods. Okay, what else can we do with these? We can fine-tune them to predict binding sites. And this is something I'll talk about more in depth towards the end when I talk about LoRA and QLoRA. But the idea here is we can fine-tune this as like a binary token classification task and set, say, 0 to non-binding sites and 1 to binding sites, and just train with a regression head on top, and get a model that tells us where there are likely to be binding sites in a protein sequence just based on the sequence alone. And once we have this, we can use this and use this in a model for generating proteins that bind to a specific protein. And there's a couple of ways that we can do this. One approach that some people have tried is just fine-tuning ESM2 using the masked language modeling objective on concatenated pairs of proteins and masking out one of the proteins completely so that the model learns to generate proteins that bind to a target. And the efficacy of this approach is still kind of to be determined. I'm experimenting with this along with some other people to see how well this actually works. It does seem to work, and the people that actually came up with this idea are reporting that they have really good hit rates, and that they may be actually getting higher hit rates than RF diffusion, which is kind of surprising to me because RF diffusion is actually a very good model. So we can fine tune it in this way, or we could use ESM2 as the backbone in a diffusion model. And so I'll talk a little bit about diffusion models. I won't spend too much time on that, but this is just an image of RF diffusion generating a protein. And this is actually a protein complex that it's generating here. So this is a diffusion model that generates proteins, and we can use ESM2 as the backbone in one of these diffusion models and generate proteins this way as well. So there's very little work on this. There's one model that I'm aware of that uses ESM2 as the backbone as a diffusion model. And it's not well tested yet. So working on this would be a really great project for someone if you're interested in diffusion models and also in language models. This might be a good project for people. It would be more similar to EvoDiff than RF diffusion, I think, because you would probably want to do sequence level diffusion instead of 3D diffusion in like backbone coordinate space. And this can be used for things like scaffolding functional motifs. So let's say you have like a region of a protein that you like that has a certain function. Maybe it binds to a target protein that you're interested in or something like that. You can take this fragment or this motif from the protein, and you can scaffold it with these diffusion models and build a protein out around this little motif that holds it in place and maintains its functioning. You can also use diffusion models to generate binding partners to target proteins. And this is kind of what I was getting at with the binding site prediction. So for example, for RF diffusion, you can give RF diffusion binding sites. So once you've fine-tuned your ESM model, to predict binding sites, you can use those binding sites in the diffusion model and tell the diffusion model how you want to generate a binder for your target protein, like what regions are going to be good for binding sites and where it should diffuse. And once we have this, and we have our binding partners generated, we might want to optimize them. So the next thing that you can do is you can use ESM2 as an expert in EvoProtGrad. EvoProtGrad is gradient-based discrete Markov chain Monte Carlo method that performs something called in silico directed evolution. So you're doing the directed evolution on your computer, not in a lab, to cut back on lab costs. And essentially what it does is it takes gradients of the model and uses the gradients of the model to propose good mutations for the proteins. And once you have a proposal for good mutations for the proteins, we mutate the protein, and we do that iteratively over and over again to try and get better and better mutations of the protein. And we use the ESM2 model as like a scoring method, similar to what I was talking about to begin with. And we get an evolved protein that has hopefully a higher score than the wild type protein and has higher functioning and a better fitness than the original wild type protein. And so we generate our binders and then we evolve them to optimize them if they're not binding well, using EvoProtGrad. And then the next thing that we can do is we can fine-tune ESM2 to predict protein-protein interactions. So one way that we can do this that I'm working on currently is concatenating pairs of proteins, randomly masking out some percentage of the residues for the proteins that we know interact with each other, just using the MLM objective. And pairs with a lower MLM loss have a higher likelihood of interacting in this case. And the trick here is when you cluster your proteins, you need to cluster them in a way so that you can create a train test split that is good for protein-protein interactions, which is actually really hard to do. And the reason for this is when you have pairs of interacting proteins, you want your train set and your test set to not have any overlap. You don't want sequences in your training set that are similar to sequences in your test set. And so I came up with a new way to cluster protein-protein complexes using something called persistent homology, which I can discuss more in depth a little later. But the idea is you compute the embeddings for each protein, and then you have a summary, a sort of geometric or topological summary of those embeddings for each protein through something called a persistence diagram. And then you compute the similarity of the persistence diagrams, which is like a summary for each sequence. And using the similarity between those persistence diagrams, you can cluster your protein-protein complexes in a way that gives you a good train test split where the test set is not similar to the training set and you don't get overfitting. This is actually a really common problem in biological data. When people move into the biological space, there are a lot of these little nuances that people don't realize where you have to create train test splits based on like sequence similarity or based on protein families to prevent overfitting, because you'll get data leakage if you don't do this. So you can think of it as like sequence similarity is like you have two sequences that are, I guess the best way to explain it would be using like Hamming distance or edit distance. If their edit distance or Hamming distance is small, you probably don't want them to be separate in the train and test split. You want them to be either both in the test, excuse me, both in the test set or both in the training set. And so finding a new way to cluster proteins using protein language models and to cluster protein-protein complexes using protein language models is actually really beneficial for this task. And there's a few other ways that we can fine-tune ESM2 for protein-protein complexes. So there's a few models that have come out recently that do a different method. Microsoft recently came out with a LoRA model that fine-tunes to predict protein-protein interactions. And there's also Sense PPI, which I'm in the process of testing out now, which seems like a pretty good model for this. And they use the embeddings from ESM2 in something called a Siamese network, which I don't have a lot of familiarity with at the moment, so I can't answer a lot of questions on that, but it seems to be a pretty good model. So the idea here is we create a pipeline for creating binders. We fine-tune ESM2 or use a different model like AF2bind or something like that to predict binding sites. Once we have our binding sites, we can use those in a diffusion model to generate binders for a target protein. Then once we have our generated binders, we can optimize them using EvoProtGrad with ESM2 as the expert. And then once we've optimized our binders, we can predict binding affinity with ESM2 or some other model that uses ESM2, for example. And then finally, we can test in the lab. And the idea here is that all of these steps before testing in the lab are supposed to save you lab work and prevent you from accruing like large costs in a wet lab, having to test binders a lot. And so this could drastically cut back on lab costs if we set the pipeline up correctly. And I won't talk about this too much, but if you're familiar with Shannon entropy and perplexity, so I'll go through these quickly because I feel like everybody here is probably familiar with Shannon entropy and perplexity. But the point is, perplexity is the exponential of Shannon entropy. And if you assume the uniform distribution, you get this formula for perplexity. And this is for autoregressive models. But we're working with ESM2, which is not an autoregressive model. It is a bidirectional model, more like BERT or Roberta. So we don't really have perplexity for the model. We have pseudo perplexity, which is a little different. But what we do have, excuse me, what we do have is we have intrinsic dimension. So intrinsic dimension in recent research has been shown to predict perplexity. It's actually highly correlated with perplexity. And so you can use it as like a stand-in for perplexity. And that can give you a lot of different things. So you can use that for a lot of interesting things that I want to talk about next. So the idea here is we take the embeddings from ESM2 for a protein sequence. And then we use a method like persistent homology or two nearest neighbors or MLE or something like that to estimate the intrinsic dimension of the manifolds that the embeddings lie on. And once we have estimated the intrinsic dimension of this manifold that the embeddings are supposed to lie on, we can use this to measure the complexity of a protein. So this is like a measure of complexity for individual proteins. And what's really interesting that I've found is that when we perform directed evolution on the proteins, we actually end up increasing the intrinsic dimension. So intrinsic dimension increases when you're performing in silico directed evolution. And I'm still not sure exactly how to explain that or why that's happening, but it seems like evolved proteins are a little more complicated than natural proteins or AI-generated proteins. And so the idea occurred to me because intrinsic dimension also predicts ease of adaptation to a particular task, maybe we should use this in a curriculum learning strategy. So there's research showing that if you if you measure the intrinsic dimension of, say, a text in NLP, this is going to give you an idea of how easy it's going to be to adapt your model to that data set. And so the same thing should hold for proteins. And if we order our data set in terms of intrinsic dimension, where we train on lower intrinsic dimension first, and then progressively higher and higher intrinsic dimension proteins, we should get a good curriculum learning strategy where the model has much smoother training curves, for example, much smoother loss curves, much more stable training than we would if we just randomly, selected proteins at random to train on. And so this idea can also be used in NLP and vision. If you compute the intrinsic dimension of your text and then order your data in terms of intrinsic dimension, you should be able to get smoother loss curves and more stable training. And the other thing that this can be used for is potentially detecting AI-generated proteins, which seems to be interesting to some people. So the method was originally developed to detect AI-generated text in NLP. And then I adapted it to the protein area. And it turns out that AI-generated proteins from a model like RF diffusion or EvoDiff tend to have lower intrinsic dimension than natural proteins. And those tend to have lower intrinsic dimension than evolved proteins, where we evolve them using EvoProtGrad. And so in theory, you would order your proteins in terms of which ones are AI generated, because those are likely to have lower intrinsic dimension. And then you would train on higher intrinsic dimension natural proteins. And then you would train on evolved proteins that had higher intrinsic dimension still. And like I said, this could be transferred to NLP. There's no reason you can't apply the same method to train on natural language text. And like I said, the original method was also developed for NLP anyway. So a lot of these ideas transfer over if that seems like interesting to you. The other thing that the intrinsic dimension can be used for is determining which layers of the model are going to be good for certain downstream tasks. And so if you look at the way the intrinsic dimension evolves throughout the model, this is a graph for 3 different sizes of ESM2. And the layers around 0.6 to 0.8 are going to be where the most geometric compression happens. So we have a lot of geometric compression happening around 0.6 to 0.8, where there's this nice flat plateau. And there's recent research that came out that shows that these are the layers that you're going to want to use for things like remote homology detection or sequence similarity searches. And let's see. Yeah, so like I said, we can use protein language models to cluster proteins or protein-protein complexes by getting the embeddings of the proteins or protein-protein complexes and then using persistent homology to cluster them. And once we've clustered the sequences, we can use this to actually replace multiple sequence alignment. So there's a recent method that came out known as VC-MSA, which is vector clustering MSA. And it basically shows how you can completely replace multiple sequence alignment with a protein language model. And I'm sort of building on top of that using this new clustering method that in their method, they get the embeddings, they compute the average of the embeddings, and then just compute cosine similarity between the averages of the embeddings for each protein to get sequence level clustering of the proteins. And I'm saying maybe we should do that a little bit differently to have something that's a little more robust in terms of sequence similarity searches using persistent homology. And let's see. Right, and so we can also use embeddings from ESM2 and structural models. If you're interested in structural models, there's some recent research out that shows that including embeddings as feature vectors in like graph neural networks or graph attention networks or graph convolutional neural networks, including the embeddings as feature vectors actually dramatically improves the model's performance. And there's other research showing that the larger you scale your models, the less the structural model actually helps. They did some ablation studies where they tried to figure out like what parts of the model were actually contributing. And it turns out the protein language model, as you scale and get to larger and larger protein language models, ends up being the most important part of the model. Okay, so let's talk about sequence and token classification tasks and fine-tuning with LoRA. So I'm going to get into LoRA in just a minute, but there's a lot of different fine-tuning cases that you can work with for protein language models. If you're familiar with biology at all, gene ontology is basically a way of labeling sequences with a function. So we can predict the function of a protein using protein language models. We can predict where the protein is naturally occurring. So like subcellular localization tells you where the protein occurs. We can predict what family the protein is in. We can also do token level classification tasks and predict binding sites, post-translational modification, and secondary structure, and a bunch of other things that I don't even have time to mention. But suffice it to say, any kind of sequence level classification task or token level classification task that you could cook up, you could probably fine tune ESM2 for it. Okay, and then let's talk about LoRA a little bit. So LoRA, I'm sure a lot of you are already familiar with this from doing NLP stuff, but I'll just mention it just in case there are some people who aren't familiar with LoRA. So we take the original weight matrix of the model, or original weight matrices, I should say, plural. We freeze them. Then we add in a rank decomposition pair of matrices that that we train on top of the original model that's not frozen, and those those are much more efficient to train than the original model, because when we choose a rank in the middle for these two weight matrices, we choose it such that we're reducing the parameter count in the model. So if you I have a couple of pictures here that kind of illustrate this concept. So in the next slide, if we look on the left side, we have the frozen weight matrix of the original model, and on the right side we have the LoRA. And if you just look at the raw parameter count here, the parameter count is actually the same. And so for a LoRA like this, we wouldn't actually have any benefit at all, because the number of parameters on the left side and the number of parameters on the right side are actually the same. So in this case, though, if we look at this slide, the left side has 100 parameters and the right side only has 40 parameters. So we've got a 60% reduction in parameters. And this reduction is usually much more dramatic for actual weight matrices in models. When you're working with like thousands of dimensions in a transformer model, and you choose a rank that's around like 10 or 12 or something like that, you're going to get a huge reduction in parameter count. And then QLoRA just quantizes this whole process and gives you an even more compute efficient way of training your model, because we're working with lower precision parameters. And so we get an even more drastic reduction in how much compute we need to train these models. And so you can train really big models with LoRA and QLoRA that you on very limited modest hardware. So I did all of these on a single H100 for very large models. So some of the models that I trained actually were in the billions of parameters, and you don't really need an H100. Like, you could do this on a small GPU with like 24 gigabytes of memory. You just have to use a lot smaller batch sizes, and so your training stability probably wouldn't be as good. You probably get much noisier loss curves and things like that. But you can train really big models on really modest hardware, and in some cases you can actually get better performance. So for some of the models that I've trained, I got slightly better performance with LoRA than I got with full fine tuning. And part of the reason for that is because of overfitting. So something that I discovered was that LoRA and QLoRA actually serve as a really important regularization technique. Exactly why that's true is still kind of a mystery to me. I think it has something to do with geometric compression and the way that the LoRA reduces the intrinsic dimensionality of the data. So if you choose a rank that's lower than the intrinsic dimension of the data that you're training on, you're forcing more geometric compression onto the model, and it's forcing it to find a lower dimensional space to sort of model these things in, and it ends up serving as a kind of regularization. And if you look at this this chart here, I have it in graph form as well. The idea is, if we look at the differences between the red and the blue and compare them to the differences between the gray and the tan, we can see the differences between the gray and the tan are actually smaller. So this is describing the differences between the train and the test metrics for adding a LoRa to the query, key, and value weight matrices versus adding LoRa to all weight matrices of the model. And if I add in a LoRa to all of the weight matrices of the model, I actually get less overfitting than if I adapt fewer weight matrices. And so this is showing me that the more I adapt the model with a LoRa, the more weight matrices that I adapt with a LoRa, the less overfitting I get. And if we compare that to full fine-tuning, we see the differences are actually much worse for full fine-tuning. And I've got that in a table as well. So here are the differences for full fine-tuning on the left, query, key, and value in the middle, and then all weight matrices on the right. And if you notice, like each time I move over to a new column, the differences are cut in half, basically. And so if I adapt the entire model, all the weight matrices with a LoRa or a QLoRA, I actually get much less overfitting than I get with full fine-tuning. And I found this really interesting, and especially for biological data, because overfitting on biological data is such a huge problem. And you're always having to consider things like sequence similarity when you're creating your train and test split. And to have something that helps regularize and helps reduce overfitting, and that can also, like, sometimes if you do it just right, give you better performance than full fine-tuning, I think is like really interesting, a really interesting find. And there's also recent work out by Microsoft where they train a LoRa for protein-protein interaction prediction, and they actually got better performance with LoRa than they did with full fine-tuning as well, which I found really interesting. And with that, I think I'm gonna wrap things up and leave some time for questions. That's pretty much the end of my slides. So yeah, I'm gonna wrap things up and leave some time for questions.
Okay, then thank you very much for this interesting talk. I'm very happy that I recorded it because I definitely have to rewatch some sections again to understand everything. Yeah, I will just allow everybody to unmute themselves as well, so they can ask questions, and then yes, now everybody should be able to unmute themselves if they want to ask questions. We had one question in the chat. I don't know if Olivia want to ask herself. Otherwise, I can just read out her question.
Yeah, you can read it. I think, or let.
Hi. Yeah, thank you so much
Okay.
for oh, sorry.
No, go ahead. I'm
Oh,
listening.
thank you so much for your talk. I was kind of wondering, when you were talking about like the Hamming distance, I was kind of wondering what metric is like, or what threshold would be too small to for separating them into the training or the test set.
Yeah, so that depends. I think that depends on what model you're using, what task you're training for, and things like that. So like for when they trained ESM2, I believe they did it on UniRef 50, which is 50% sequence similarity. And so and then when they created their train and test split, I'm not exactly sure what the sequence similarity difference there is, but they split it so that the similarity between the test set and the data set isn't too high. And honestly, that I'm not sure what the percentage difference there was. So I'm trying to think of if I can remember. I would have to look it up. I'm not sure what the numbers are for like what threshold you would want to choose for sequence similarity for creating your train and test split. But ideally, you would have your test data set be very dissimilar from your training data set in terms of sequence similarity, or something like sequence similarity. So for some models, you might also want to consider structural similarity instead of sequence similarity, because there are proteins that are structurally very similar, but have very low sequence similarity, which to me kind of indicates that sequence similarity is maybe like a bad measure of similarity in some cases. And so that's why I was trying to figure out a new way to cluster like proteins and protein-protein complexes using protein language models and their embeddings. Because if you can compute a kind of similarity that is known to encode some of the structural information as well, you could probably get a better train test split than if you just use vanilla sequence similarity methods.
Gotcha. Thank you. And I also had another question about training based on the intrinsic dimension.
Yeah.
So for that method, would you train for a couple of epochs on a lower intrinsic dimension, and then like keep I guess like training for a different number of epochs based on the intrinsic dimension? Or
So I would just order the data in terms of intrinsic dimension to create a curriculum learning strategy for the model. And the number of epochs that you train, I guess, that could complicate things. But a lot of the models that I was training, I was just training for a single epoch. So yeah, I would say order it in such a way that you train on simpler sequences first and then train on more complicated sequences later.
gotcha. Thank you so much.
Are there any other questions from the audience?
Yes, I was also curious as to whether or not you had any conjectures for why AI-generated proteins had a lower intrinsic dimension. Just kind of curious about that.
Yeah, that's a really good question. So the idea comes from NLP. The method was originally developed for NLP in detecting AI-generated text. And it was shown that at least in this paper that I pulled this idea from, it was shown that AI generated text in general has lower intrinsic dimension. And I think they tested on GPT 3.5 and not on GPT-4. And so like I would be interested to know like how that works for different models and how well that actually holds for different kinds of models. I've only tested this on a small number of proteins where I generated a protein using RF diffusion and then got a sequence for that protein using ESMFold, which is an inverse folding model. So you can get a sequence that folds to the 3D backbone that RF diffusion generates using an inverse folding model like ESMFold. And once you have that, you can compute the intrinsic dimension of that sequence using ESM2. And the pattern that I was seeing was that AI generated proteins had slightly lower intrinsic dimension than natural proteins that you might just pull off of like UniProt, for example. And the evolved proteins, the ones that you perform directed evolution on, have even higher intrinsic dimension than natural proteins. And why that would be the case is still a bit of a mystery to me, but the intrinsic dimension is known to correlate with the perplexity. So this would imply that the perplexity also goes up. For natural proteins, you should have a slightly higher perplexity than AI-generated proteins. And for directed evolution proteins, you should have even higher perplexity for them because they should be more complicated and a little more confusing to the model. And again, like why this is happening is still a bit of a mystery, but it does seem to be happening for sure.
Okay, so it's fair to say that you think it might be a component of a given model itself, as opposed to, say, maybe how the model goes about optimizing for the creation of either, say, text in the case of NLP or proteins in case of biological work.
Yeah, and my guess is you can probably, like, if you were to do this with NLP, and you were to use like GPT-4, for example, my guess is you could probably, with like some kind of prompting, get out text that is higher intrinsic dimension than just your average text that you might find on Twitter or something. And so I don't think it's a super like straightforward, easy way to differentiate AI generated text from AI or from natural text, because I do believe like better models are going to model language or proteins for that matter more accurately. And so as the models improve, the intrinsic dimension gap is going to be lower. And this is just a guess, but I think if you use the curriculum learning strategy where you train on lower intrinsic dimension first, and then progressively get higher and higher intrinsic dimension proteins that you're training on, you might get a model where that gap is even lower, because the model has like understood more and has encoded those proteins in a way that it compresses things better. So yeah, that's just a guess though. I'm still a bit confused as to why this is happening, but it definitely is happening.
Well, that makes sense. The explanation you provided does make a lot of sense. Thank you for clarifying.
Yeah.
We also have an idea, for example, because the intrinsic dimension can also vary strongly between different models. So if you train a model on some other sort of data set, it will capture different information than it is trained on another data set. Do you think that the ESM embeddings are especially suitable for this kind of analysis that goes into another pipeline, or where do you see differences between models?
Yeah, so you definitely see differences between models, and I'll just go back to, let's see, this one here. So this is for 3 different ESM2 models, and this is just the, here they use the maximum intrinsic dimension of a data set, and they look at how the intrinsic dimension evolves throughout the model. And you can see that the same kind of pattern emerges, and they've done this with other protein language models as well, and a similar pattern emerges where you get this like peak in the beginning, and then things drop off and plateau, and then they grow again towards the end. And this actually happens for other protein language models, not just ESM2. And it happens for different sizes of protein language models, which is really interesting. And if you look at the same sort of graph for vision models and language models, they have a different shape to them, but they all have a similar sort of shape if you compare them like within their domain. So like if I compare vision models to other vision models, I'll get a similar intrinsic dimension profile through the layers of the model. And if I compare language models to language models for like NLP, I'll get a similar sort of curve. It looks different from the protein language models, but it's similar for other NLP models, I should say. So it seems to vary depending on what domain you're working with, what kind of data. So if I'm working with vision data, I'll get one kind of profile. If I'm working with natural language data, I'll get a different kind of profile. And if I'm working with protein data, I'll get a third kind of profile that looks like this. And why, again, like why they should be doing this and why it changes from one domain to the other is very much a mystery. They don't say in the literature why this is happening. They don't have any conjectures about why the profiles look the way that they do. But my guess is you could probably use, for proteins, you could probably use another protein language model, and because the profile of the intrinsic dimension is going to be very similar, you would probably get very similar results for intrinsic dimension of proteins. I don't think it would vary a lot between models. So I don't know if ESM2 is like especially good for this over other protein language models. I think you could probably use another protein language model and get very similar results.
Okay.
Yeah, I think that's part of the reason why I was a bit skeptical initially about it being the model. Domain makes more sense, what you described.
Yeah.
Any other questions? Okay, lastly, then I will ask one more question. You mentioned that you work on this new clustering technique in the beginning, where you use persistent homology and ESM2 together. Maybe it's just my not knowing things, but could you maybe explain what persistent homology is and why it is especially suitable for proteins and how it works together with ESM2?
Yeah, so persistent homology is just a clustering method. It's kind of you can think of it as like a fancy dbScan. So if you're familiar with dbScan, it's essentially dbScan at multiple scales. So you essentially you run a dbScan for a whole range of epsilon values, and you for each epsilon value that you fix, you get a different set of clusters. And there's a diagram that sort of summarizes the different kinds of clusters that you get for all the different scales of epsilon for the distance in dbScan. So it's also kind of similar to HDBScan if you're familiar with the hierarchical clustering that they use for HDBScan. It's very similar to that as well. You can get a diagram that sort of summarizes this data. So if I were to do this for like embeddings for a protein, say, so I give my ESM2 model a protein, and it gives me back some embeddings. And those embeddings in theory lie on some kind of manifold. I can do a dbScan at multiple scales and get different kinds of clusters at different scales. And then I can summarize this in a persistence diagram. And then you can use those persistence diagrams to cluster the proteins, because that gives you a sequence level summary of what the protein is in terms of the embeddings. And I can compute things like L2 distance between like something like persistence landscapes, or I can compute like Wasserstein distance between persistence diagrams. And I can cluster the proteins in this way.
Okay, and why is this superior to some other methods? Like, I don't know, I don't even know what other methods there exist in this space, but is there something about the centroids of the clusters, or does it have to do with, like, for example, why is it better than something very simple than k-means or something like this?
Yeah. So I think like you're getting a more robust description of the embeddings. So the other method that I've come across that people have used is computing the embeddings and then just taking the average of all of the embeddings, and then computing like cosine similarity between the averages. So if I have like embeddings from two different proteins, I compute their averages, and then I compute the cosine similarity between those two averages. I get some description of how similar those two proteins are. And like how this compares to the persistent homology method is basically, instead of taking the average, you're maintaining all of the embeddings, all of the information in the embeddings. You don't take the average. You take sort of a topological descriptor of all of the embeddings at the same time. And this, like, I think it retains more information. And so you get a more nuanced description of how similar the proteins are.
That makes sense. Okay. Very nice. So if there aren't any other questions, we are at 1 hour sharp. Yeah, please.
Yep, just one more question.
Yeah, sure.
So given the nature of how you're applying persistent homology in this context, do you think that a similar approach could be applied for NLP, for better capturing, say, the nuance of certain text embeddings?
If you just wanted to compare like two different texts and see how similar they are, I don't know. I've tried this some, and like I feel like I'm just not as good at NLP as I am with proteins. And so I don't have a strong intuition for this. I have tried it some to see like if you can compare two texts using this sort of method. And I guess like in theory you can, but I don't know I don't know enough about NLP to really say for sure how well it would work. But like you would hopefully get a comparison that tells you how similar they are, like in terms of like content, or in terms of like what sort of topics they're talking about, or something like that. And I guess in theory it could work, but I don't really know. I honestly I can't really say much about it, because I feel like my intuitions for NLP are just not as strong as they are for proteins.
That's fair. Thank you.
But I think, like, given the sequential nature of both of these tasks, and if you interpret the amino acid as like the vocabulary where you can model, a lot of things work between those areas, and you can apply a lot of classic NLP methods to the biology world. So this is definitely an interesting approach.
Yeah.
Okay, then again, we're at 1 hour very sharp now. Thank you very much for your time, and thank you for presenting. I will edit this video, put everything you mentioned in the description, and then we'll share it with all of you to rewatch the presentation and discussion.
Awesome. Thank you.
Yeah, thank you everyone for attending.